Hello students, I am Akshay Deshpande and welcome to another class of biology. And today we will be beginning with a new chapter from the cell unit, that is the cell division and cell cycle. Okay, and this is a short chapter. Okay, and it is not quite mentioning for you people that you have not studied about cell division and cell cycle. If you hear the term biology, if you have been studying biology since your school days, right, so you can assume that cell division and cell cycle is one of the most repeated chapter or a concept in the biology. Okay, we can say it is one of the fundamental aspect, especially the cell, the unit of life, all the contents of the cell, that is organelles and cell division. Okay, I don't think that apart from these two chapters, which are the most repeated chapters, I believe, okay, none of the other chapters are that much repeated, okay, in the whole biology what you people study. Okay, so fairly you people will be having a very good amount of idea about the cell division and cell cycle. Okay, but cell cycle you people are studying it for the first time. But however, we can say that about the cell division you people have studied about it quite a lot of time. Okay, so we will be starting our discussion mainly on the cell cycle today, concentrating mainly about the interphase. What is actually interphase and what does interphase is mainly involved in all of the whole cell cycle. Okay, yeah. So now let us look into some of the important characteristics of the cell. Okay, see, we know that the growth and reproduction are the characteristics of the cell, right? So not only growth and reproduction, we also even consider the metabolism, right? Even the metabolism are some of the important characteristics. You people will be studying about the characteristics of the cell more elaborately in the first chapter, that is the living world, right? So in the first chapter you people will try to tell what is living and what is non-living, right? On what basis you call it as a organism, as a living or non-living, right? And the same points that we have mentioned, that is the growth and the reproduction and also metabolism. Okay, growth, reproduction, metabolism, these are some of the defining feature of the living organisms. Okay, and as we all know, cells are ultimately living organisms, so hence they do have the ability to reproduce and also to grow in size. Okay, so in simplest terms, what do you mean by cell division? Okay, a cell division is a division of a single cell to divide into two daughter cells. Okay, now this is what we mean by the term cell division. Okay, division of a single cell into two daughter cell is called as the cell division. Okay, now the daughter cells, if we have to draw this diagrammatically, right, we are having one cell and this will divide into two daughter cells, right? This is how we can show it diagrammatically, right? Now these cells are not immediately ready, right? They are not immediately ready, right? They are not immediately ready to divide, right? They are not immediately ready to divide, right? They require some time, right? And this time is mainly used for one important purpose, and that is the growth. Okay, it is mainly used for the growth purpose. And after growing for a certain particular period of time, then they go for what? Again for the cell division. They will go for again for the cell division. Exactly, right? Again from this cell we will get another two daughter cells. Again they will get two daughter cells. From this cell also you will get two daughter cells, right? So in this way the process repeats again and again, right? So here we have mentioned that the daughter cells actually grow for a certain period of time. Okay, and the growth mainly includes accumulation of nutrients. Okay, the growth mainly includes the accumulation. Okay, it is mainly the accumulation of the nutrients. Yes, ultimately nutrients are required, right? The nutrients may be in the form of the proteins, right? okay, maybe in the form of proteins, okay, it may be in the form of the energy, that is the ATP, right? Ultimately, the cell division does require some amount of energy also, right? And also the enzymes that are required for the various processes that will be occurring during the cell division, right? And these enzymes are none other than proteinaceous in nature, that we have discussed in the last class. So we can say that growth mainly is including the accumulation of nutrients, and these nutrients mainly help in the cell division process, right? So the daughter cells will grow, and when it is ready, then only it will go for the division process, that is the cell division process. Okay, so what is the ultimate result of the cell division? The ultimate result is that the population actually increases. Okay, so now here we are having two important concepts, that is the reproduction, okay, reproduction and increase, okay, increase in the cell number, right? Increase in the cell number, we can say increase in the cell number, or even term reproduction. Okay, it is the term reproduction. Now we can say that both of these two terms, that is the reproduction and increase in the cell number, or cell density, right? Cell density. So density means mainly increase in the number of the cells. It is called as cell density. So both of these two terms are synonymous, or we can say that both of these two terms are actually equivalent in nature. There is no difference between reproduction or cell density. Okay, but however, these two terms can make some sense only if it is going to be in the case of unicellular organisms. Okay, only in the case of unicellular organisms you can say that reproduction and increase in the cell number, or increase in the cell population, both mean the same. But however, in the case of multicellular organisms, you cannot say. Okay, you cannot say that reproduction and the cell density is same. Both of them are actually different. Okay, in the case of multicellular organisms only, right? So increase in the cell number, that is mainly due to the cell division process, right? So now the cell growth and cell division mainly allows the cells to grow. Okay, so that is what we have mentioned the above point, that the cell growth and cell division allows the cells to grow and divide to form millions of cells. Divides to form the millions of cells, right? See, in the case of multicellular organisms, right? In the case of multicellular organisms, you can notice that they are made of, okay, they are mainly made up of millions of cells, right? In the case of humans, we are made of millions of cells, right? So new cells are constantly being added. The new cells are constantly being added, that is mainly what we can say due to the cell division process. It is mainly because of what? The cell division process only. Without cell division, there will be no formation of millions of cells, and we can say that there cannot be any kind of multicellular organisms, right? But however, in the case of the unicellular organisms, the increase in the cell number and also reproduction, both can be achieved by the cell division itself. Both can be achieved by the cell division itself. This is what we mean by the cell division, and why cell division is more important. But we are having one more term that is called as a cell cycle. Okay, see, generally if we are using any term a cycle, okay, now what do we mean by cycle? Okay, now cycle in the sense any process, okay, it is any process that basically starts from one point, okay, starts from one point and ends or comes back to the same point again, okay, to the same point again. Okay, now such kind of process is called as a cycle, or we can say that they are actually the cyclic process. They are called as the cyclic process. For example, we are having assume that we are having a molecule starts from to B molecule. The B molecule next goes to C molecule. C will result in the formation of D molecule. From D to E molecule, and E to back to again A, and again from A to B, B to C, C to D, D to E and A, and this cycle, this process goes on in a circular fashion, in a repeating event again and again and again and again, right? Now such kind of process we generally refer them as the cyclic process. Okay, so hence we can say that cell cycle, in the case of cell cycle, it is a cyclic process. It is basically a cyclic process. Okay, it will be repeating again after and after. Okay. Now, the same way over here, the cell division is an important process. Okay, and now you can see that during the cell cycle, the DNA replication and the cell growth actually takes place. Okay, so the difference between cell division and the cell cycle is that the main goal of the cell cycle is, the cell cycle is mainly involved in informing, it is mainly informing about, okay, it is mainly informing about the events that occur during the cell cycle, right? So the cell cycle mainly tells, it mainly informs us about the events that they occur during the cell cycle, right? These events, the most important term is what? The events. Okay, now these events are actually biochemical events. Okay, biochemical events, right? And interestingly, these biochemical events are not occurring randomly, right? It is not occurring in a random fashion, but however, it is occurring in a very, very orchestrated, or we can say in a series of events. Okay, in a series of reactions. Okay, not, we can say, actually they are reactions, ultimately they are reactions only, but reaction can be a very specific term, but we can use that series of events. Okay, now events are more general term. Okay, we can use events, can be a more general term for our discussion, but reaction kind of a very specific word we can use for here, right? So here we are actually having the definition for the cell cycle: the sequence of events by which the cell duplicates its genome, synthesizes other constituents, and eventually divides into two daughter cells. Okay, that is called as the cell cycle, right? So now here, if you observe this particular statement, we have mentioned about the biochemical events that are occurring in the cell cycle, right? So here we mentioned about the biochemical events, right? And also the definition gives us an idea regarding these biochemical events, and these biochemical events are none other than about the duplication of the genome, synthesis of other constituents that are required for the cell division, and finally the cell division process itself, right? So the three important event that you people can observe in the biochemical process, which is a biochemical process, is that it is DNA duplication or DNA replication. Okay, I will just DNA replication, and next is growth, and finally growth, yes, exactly, synthesizes the other nutrients for cell division, which is none other than growth itself, right? And finally we are having the cell division itself, right? And these three events are actually biochemical in nature. Okay, they are biochemical in nature because DNA replication, it involves a biomolecule called as DNA. Growth, synthesis of nutrients, and nutrients in the cells are none other than the biomolecules, right? And finally cell division, dividing the cell contents, is again none other than dividing the cell constituents itself. cell itself. Hence we call it as the biochemical events. The sequence by which the cell duplicates its genome, synthesizes other nutrients, and eventually divides into two daughter cells is called as a cell cycle. This is a definition for the cell cycle. Now, the cell growth may be observed during some other times, but however, the DNA is replicated only during the cell division, or we can say only during the cell cycle. Now, this is a very interesting point that we have to notice, that the cell division can occur at any moment of time. Okay, but however, you can see that during the whole process of cell division, or during the whole process of a cell cycle, the DNA is replicated only once for all and only at one particular moment of time. During the any other part of the cell cycle, or during the any stage of the cell cycle, the DNA duplication or DNA replication does not occur once again. Right? Now, this is mainly because of a special kind of molecules. They are called as the helicases. Right? It is called as the helicase. Now, I don't have to tell about the every specific helicase that is involved in this process, but remember, helicase are the enzymes that are mainly involved in unwinding the DNA. Okay? So, adding the helicase molecule to the DNA is a confirmation that the cell is going to divide, and hence the genome or the DNA should be replicated or it should be duplicated. That is the message it sends. But soon after duplication, when the helicase is removed, that is an indication that there will not, or there is not going to be another round of duplication, right? So in the case of cells, you see that there are different types of helicases, okay? And there is one specific helicase which makes sure, which ensures that the duplication of the genome occurs only once in a cell cycle. Okay, once in a cell division. It is not repeated again. Now that is ensured by one special kind of helicase. Okay, if you want, I can tell you the name. It is called as the MCM helicase. Okay, it is called as the MCM helicase. I don't remember the full form of the MCM, but the name is actually the MCM helicase, and you people can just check out the meaning of or full form of the MCM helicase, right? So cell growth can be observed at any moment of time or at any other times, but the replication of the DNA is observed only at one particular moment of the cell division and none other than that. Now, the chromosomes are finally divided and are distributed to the daughter cells. Now, this is also a very important event that you people observe, that is the result of cell division, right? So, here, if there is one cell, it divides into two daughter cell. It means that this particular daughter cell and this particular daughter cell needs to have all the required genetic material or all the genes that this parent cell actually had. Okay, so that even this cell can faithfully replicate its genome and distribute it to its own daughter cells. So, missing of any part of the DNA can ultimately lead to disastrous consequence. Okay, deletion or duplication or missing of any part of the DNA will ultimately lead to disaster consequence. What may be the consequence? What kind of disaster consequence I may be speaking of? It may be cell death ultimately, right? If okay, it may be cell death, or it may be cancer in some cases. In worst cases, it may be turning into cancer, right? So hence, the chromosome should be split, or the DNA part should be divided and distributed equally to the daughter cells. So this is the result of the cell division. Okay, so what is the ultimatum? What is the intention behind the cell division? The intention behind the cell division is not only increasing the number of cells, but at the same time distributing the genetic content or the genetic material equally to the daughter cells. That is also one of the important reason behind the cell division. Now moving on, let us look into the different phases of the cell cycle. What are the different phases or the categories or stages of the cell cycle? You see that the first phase we are having is called as the interphase, and next is called as the M phase. The M mainly stands for the mitotic phase. It is called as the mitotic phase. It is called as the mitotic phase. And the interphase is actually thought was once upon a time, at least it was once upon a time thought that it is called as the resting phase. It was thought that it was actually the resting phase. Okay, but interestingly, it turned out that these interphase is mainly involved in synthesizing all the materials, all the molecules that are ultimately required for the M phase. Okay, and the M phase, as the name itself indicates, it is called as the mitotic phase, and the mitotic phase is none other than the classical stages that you people actually remember. That is the prophase, okay, metaphase, next is the anaphase, next is the telophase, and cytokinesis, right? All the phase that you people name it every time, it is actually the part of the cell cycle. Okay, it's actually the part of cell cycle, and before this M phase, we are having a big major phase. It is called as the interphase. And interphase was once upon a time thought to be a resting phase, but interestingly, it isn't a resting phase. Okay, all of the molecules that are required for the cell division, it is or the M phase, it is prepared in the interphase. Now you can see that we are having interesting data over here. Regarding the human cells, you can see that the human cell, one single human cell divides once in a 24-hour. Or we can say that for one cell to divide, it takes around 24 hours. Right? And but however, the same thing is not observed in the case of another eukaryote, that is the yeast. In the case of yeast, only in a case 90 minutes, that is 1.5 hours, you can see that the yeast duplicates or replicates or divides into two cells. Right? Now, out of this 24 hours, you see that the human cells spend around 95% of its time in the interphase of the cell. Now, definitely, if the human cell or if the cell is spending around 95% of time in the interphase, it means that interphase is a kind of an important phase of the cell cycle. It is an important phase of cell cycle, right? And that is the interphase. Now, why the interphase is such an important stage of the cell cycle, we shall be shortly looking into. But before that, you can see that the M phase mainly includes the karyokinesis and cytokinesis. Now, karyokinesis is splitting or dividing the genome to daughter cells. Okay, now the genome is compacted. It is compacted into a structure called as the chromosome. Right? We have discussed about the chromosomes and the structure of the chromosomes and also the classification of chromosomes in our cell unit, right? So we can see that the chromosome over here, okay, this is how a chromosome actually looks like, if I'm not wrong. Right? Yes. So what happens is that at the end of the cell cycle, you can see that one of the sister chromatid is present in one cell, and the same sister chromatid, the duplicated part, another arm, is mainly present in another cell. Okay, so you can see that the whole thing was present in only one cell, but at the end of a cell division, you can see that one is present over here, the second is present in another cell. It is present in the another cell, and this process of splitting of sister chromatids is called as the cytokinesis. Remember, students, many a times I see that the children are quite confused with the term about the homologous chromosomes and sister chromatids. Okay, so remember these two parts, okay, this one and this one, what I am marking over here, are actually the sister chromatids. Okay, they're actually called as the sister chromatids. Okay, so you can see that, remember, the chromosome is not present in this form every time. Okay, in the nucleus, you know that in the nucleus the DNA is present in the form of a thread-like structure called as the chromatin, right? Yes, it is called as the chromatin, right? And also remember that when the cell starts to plans to divide or starts to divide, before the cell actually enters into M phase, all the chromatin that is present, in other terms, all the DNA that is present, it is duplicated. Remember this point, right? So when the cell enters into the M phase, you can see that the nuclear envelope, this is called as the nuclear envelope, actually it disappears and the compaction of the chromatin starts, and as a result you see the chromosome in this particular fashion. Okay, yes, but however, if you try to assume that the content of the nucleus, that is the DNA that is present in the nucleus before the replication, okay, what is before the replication, the chromosome will be in this particular fashion. If you try to assume, okay, yes, only two arms, okay, one above arm and one below arm, it is called as the P arm and this is called as the Q arm, right? Remember this. Okay, so before the replication, if you have to compact the chromatin into a chromosome, then this is going to be the diagram of a chromosome. Okay, but however, this is the structure of the chromosome after duplication, after the duplication of DNA. Remember this. Okay, so it means that this arm that you are looking over here, okay, what I have shaded over here, this arm what I have shaded over here is actually a copy of this arm only. Okay, it is actually, this arm is a copy of this arm. Okay, yes, so I hope you might have got the point. Right? So these sister chromatids are actually the copies of each other. This 2 number what I have written is actually a copy of the 1 number. Okay, so we can say that the number of chromosomes have not increased, but the amount or the content or the volume of DNA has increased. So why the DNA volume has increased? The volume of the DNA has increased mainly because the DNA has been replicated. Now when does the DNA replicate during the cell cycle? We will be shortly discussing that. Okay, yes. So the next part over here we are having is the cytokinesis. Cytokinesis is a division. It is the division of the cytoplasm. Okay, it is mainly the division of cytoplasm. It means about the cytokinesis. Now let us look into the next, that is the interface. So now let us take a detailed look about the interface and what does the interface actually contain. Okay, so as I said earlier, once upon a time it was thought to be a resting phase, but interestingly it is not a resting phase. It is actually, we can say, a preparatory. It is actually a preparatory phase. Okay, it is a preparatory phase mainly because all the nutrients, okay, all the nutrients that, okay, are required for cell cycle or cell division are synthesized. Hence it is called as the preparatory phase. But now, as I said, it is actually the preparatory phase for the cell division. Okay, I have mentioned the same point and written it the same over here. Now, what are the different stages of the cell cycle? Okay, so now let us erase this and write the cyclic view of the cell cycle. Okay, if I have to write about the cell cycle, we can say, we can begin with the first stage called as the G1 stage. The next stage we are having is called as the S phase, and the next one we are having is the G2 stage, and the next one we are having is the M stage or M phase, and coming back to the original one, that is the G1 phase. So except this M phase, okay, except it is the M phase, you can see that the G1 phase, S phase, and the what we can say G2 phase are actually the components of the interphase. It is the stage of, or the part of the interphase. That is what we have mentioned over here. It's called as the G1 phase, S phase, and G2 phase. The G mainly stands is the gap phase. Okay, it is called as the gap phase, but however it is actually not the gap phase. Okay, as I said, once upon a time they thought that the cells were not involved in any kind of activity. Okay, but however in the case of gap phases, robust growth is actually observed. Okay, robust growth means the cell is highly metabolically active. Okay, and during this what we can say, the growth is observed. What do you mean by growth? It is mainly increase in the cell volume. Okay, you see that there is increase in the cell volume. Okay, that is why we call it as a growth, right? And why there is increase in the cell volume? Because the cell is involved. Okay, it is mainly involved in synthesis. Okay, synthesis of what? As I mentioned, all the molecules that are required for the cell cycle and cell division ultimately, right? Yes. So it mainly characterized with the growth and increase in the cell volume. The same way we are having one more phase called as the G2 phase. Even in the G2 phase, we are observing the growth and all the molecules that are required for the cell division. Okay, but there is a catch over here. There is a difference. What is the difference between the gap 1 and the gap 2 stage? Okay, in the case of the gap 1 phase, you see that all the nutrients that are produced in the G1 phase are going to be utilized in the S phase. Okay, so we can say that the G1 phase is a kind of a preparatory phase for the S phase. Okay, so whatever happens in the S phase, okay, so for the events to occur in the S phase, all the nutrient molecules or all the biomolecules required for the S phase, they are basically produced and synthesized and kept ready in the G1 phase, right? So similarly, you can see that in the cyclic representation, after the G2 phase we are having is the M phase. It means that whatever the biomolecules are required in the M phase are ultimately synthesized and kept ready in the G2 phase itself. Yes. So now, what is actually S phase? What happens in the S phase? If you ask, you can see that in the S phase, it is also called as the synthesis phase. It is called as the synthesis phase. Why it is called as synthesis phase? Because there is DNA replication. Okay, there is a DNA replication, or we can say it is a DNA duplication. The DNA is duplicated. DNA is duplicated, right? Yes, that is why just a moment ago I said that, right? The chromosome, if you have to draw a compacted form of all the chromatin or all the DNA before cell cycle, okay, before cell cycle or before cell division. Okay, before cell division, before the cell actually plans to divide, okay, or you can say before S phase. Okay, before S phase, you can see that this is how the chromosome actually looks like. Okay, but however, after the what we can say S phase, okay, after the S phase, you can see that this is how the chromosome looks like. Okay, of course, remember the chromosome is actually formed during the metaphase. Okay, but I'm just giving you an idea. Okay, an idea how the chromosome would look like if you have to compact during that is before the S phase and after the S phase. Okay, it is just an explanation over here. Okay, so now why we have actually got two sister chromatids. Here we have got one chromatid, and similarly we have got one more chromatid over here. So this, what I have drawn over here, the shaded part, lined part, this part, let me give this number 2, okay, and let me give this number as 1, okay. The 2 is actually the copy of 1 only, okay. Hence we can say that the DNA is duplicated, is actually duplicated. Yes, it is duplicated. So after duplication only, what happens? Another sister chromatid is formed or added. So we can say that the volume of DNA has increased. Okay, the volume of DNA has increased. But do you think the chromosome number has increased? No. Even before the S phase, or even before starting of the S phase, you can see it is only a single chromosome. But after the S phase, it is still a single chromosome, but the amount of DNA in this chromosome has increased. The amount in the DNA, amount in the chromosome has actually increased. Right? Yes, that is what we will be shortly explaining in the point form. Now, so let us look into the salient features of the G1 phase, which I have already told you that it is the not interval, it is the interface between the mitosis and initiation of the DNA replication. It is in between the mitosis and the initiation of the DNA replication. It's also called as the interval. Okay, interval. And next is continuously it is active and metabolically robust. Okay, and it mainly prepares all that is required for the S phase, as I mentioned earlier. The next one, in the case of S phase, the stage is where the DNA replication occurs. It is the stage where the DNA replication occurs. And as I mentioned, the amount of DNA content doubles, okay, it increases. So if I have to say, if this is 2C, then this will be 4C. Okay, I hope you people might have got the idea what I was trying to explain till this moment. Okay, if the amount of DNA, the volume of DNA, okay, I'm not speaking of number of chromosome, remember, it is the amount. Okay, if the amount of DNA in this particular chromosome is around 2C, then this it will be what, around 4C. Okay, yes. And so hence we have to note that there is no increase in the number of chromosome. There is no increase in the number of chromosomes. The next one, that is the G2 phase. As I said, all the proteins that are required for the M phase or cell division are synthesized and are produced. That is what you see in the case of the G2 phase. I hope whatever concepts and whatever points that we have discussed over here at this particular moment are quite clear to all of you. Right? Yes, I understand this particular point may be confusing for some students, but remember the number of chromosomes never increases. Okay, if we consider this as a single chromosome, this is a single chromosome, and this is also a single chromosome. Remember this. Okay, yes, but the DNA, what you can look over here, the DNA, what you can look over here, has actually increased. Okay, so the appearance of one more sister chromatid, don't you think that it is actually increasing the amount of DNA? Exactly, that is what we are trying to understand over here. Okay, yes. So now let us look into the next point, the G0 phase. So interestingly, we are having one more stage called as the G0 phase. Now G0 phase is not technically or not officially the part of the cell cycle, but we consider that some cells, if after certain rounds of cell division, okay, we can say that after certain rounds or times of cell division, okay, the cell quits. Okay, the cell quits or exits the cell cycle. Okay, or we can say cell division process, or we can say it is cell division, and enters into a quiescent stage, quiescent, okay, quiescent stage, where the cell will not divide. Okay, it basically temporarily, right? Remember, it's not a permanent stage, it is not a permanent phase. They just decide that, okay, enough, I have divided for enough period of time, but now I will stop dividing. Okay, they are just metabolically active, but they are not dividing. They never divide. Okay, at least for a moment of time, at least for a period of time. There are some cells which never divide in the human body. Okay, if I have to mention such kind of cells, for example, we are having nerve cells. Okay, nerve cells. Okay, the cells inside your in the brain, they are none other than the nerve cells, right? They never divide. Now similarly, we are having one more called as the heart cells. Okay, heart cells, that is the cardiac muscles. They never divide. Okay, they never divide in your body. Okay, now if you ask, why do nerve cells or the heart cells never divide in the human body? Because they are active throughout one's lifetime. Okay, and they are active every moment of the time. So basically we can say that these two cells, we can say, don't get, okay, they don't get time to divide. Okay, they don't get time to divide because they are active, they are highly busy cells, they are highly, what we can say, busy cells, right? Because they are active every moment of time. So hence they don't divide, and hence they are permanently present in the quiescent stage. They are metabolically active. Remember this, okay? Whatever functioning is required, they will be doing, and they are metabolically active, but they just don't divide, right? But on the contrary, some of the cells stop dividing for a momentarily or for a moment of time, but they can get back to the cell cycle phases, okay? So such cells, we are having some examples. For example, stem cells. Okay, for example, stem cells. If I have to pick, or if I have to point out one specific type of stem cell, it is called as the hematopoietic stem cells. Okay, if I have to pick one interesting kind of stem cell, it is called as the hematopoietic stem cell. Okay, the function of the hematopoietic stem cell is mainly to produce, okay, to mainly produce the blood cells. It mainly produces the blood cells, and hematopoietic stem cells are mainly present in the bone marrow. Okay, they are mainly present in the bone marrow. Remember this. Okay, so hematopoietic stem cells will not divide until there is any decrease in the blood cell concentration, until there is any requirement. Okay, so for example, don't you think the lifespan of the RBC is around 120 days? Okay, so after 120 days, what happens? Okay, the RBCs are actually replaced. Okay, the old RBCs are killed, so the replenishment, okay, generation of new RBC, production of new RBC is only possible because the hematopoietic stem cells, some of the hematopoietic stem cells will come into action, they convert into blood cells. Okay, now how that actually occurs is a completely different aspect. If possible, if we get time, we can discuss that in the human health and diseases. Okay, yeah. Anyway, but remember, stem cells are, we can say, they remain in the quiescent stage, that is the G0 phase, for a temporary moment of time. But however, whenever there is a requirement, they can come back to the active cell dividing stage. Yes. So some cells, as I have mentioned over here, some cells exit the cell cycle and enter into an inactive stage. Inactive in the sense the cell dividing property is inactive, but metabolically they are active. Okay, inactive stage called as the G0 phase, also called as the quiescent phase. Okay, yes, quiescent means mainly silent. That is what it means. But however, as I mentioned, that they are metabolically active and divide only whenever it is required to do so, or whenever there is a we can say need to come back to the cell division. Okay, so if you are thinking like what makes to come back to cell division, okay, that is mainly because of certain signaling molecules. Okay, it is mainly because of certain signaling molecules, and don't worry about these signaling molecules, it is beyond the scope of discussion in the particular class. Okay, yes. So now remember that all the animal cells divide mitotically. All the animal cells, okay, all the animal cells divide mitotically, mainly the diploid cells, okay, mainly the diploid cells, okay, and also the germ cells in the case of animals are divided by one more kind of cell division called as the meiosis. That will be another class. But however, even the haploid cells, for example, in the case of male honeybee, okay, now male honeybee is a very interesting case, because if you have to consider the development of the male honeybee, it mainly starts with the queen, okay, yes, probably you people will study about this more detail in the genetics, that is the principles of inheritance and variation. You see that in the case of a queen, what happens is the queen can lay fertilized egg, okay, she can lay fertilized egg, okay, or if she wants, she can even lay the unfertilized egg, okay, she can lay unfertilized egg, and the unfertilized egg is basically what? Developing into a male, and the fertilized egg will develop into what? A female. This is a symbol for female, and this symbol is mainly for male, okay. And we all know eggs are gametes, right? Eggs are gametes means what is the ploidy condition in the case of eggs? Nothing but what? Half of the original diploid, that is, they mainly contain N. N is an alphabet that we mainly use for what? Mainly used to tell about the ploidy condition, that is haploid condition in the case of unfertilized eggs, right? Yes. So the male honeybees are actually haploid in nature, hence the haploid cells divide mitotically. Then what about the plant cells in the case plant cells? Irrespective whether it is haploid cells or diploid cells, all of them divide mitotically. So we have discussed quite elaborately regarding the what we can say cell cycle, that is interphase. Now what we can do is we can take a moment of time to discuss about certain questions from this section what we have discussed yet. So now which of the following is a characteristic of cells? Growth, reproduction, metabolism, all of the above. We know the answer is all of the above. Metabolism, reproduction and growth, they are actually the characteristics of the cells. The next one, the two daughter cells can divide, they have to what? Grow, have energy, generate ATP, all of the above, okay. Before the two daughter cells can divide, they have to actually grow, okay. I think I missed the word before. So before the daughter cells can divide, they have to grow. So hence A is our right answer. Now which of the following event is most important in the cell cycle? Cell growth, DNA replication, cell division, all of the above. If you see that we have discussed in the first part of the class the biochemical events, okay, cell division, cell growth and DNA replication are none other than the biochemical events that we have discussed. We can say that all of the above mentioned are important for the cell cycle. D is our right answer. The next question, which of the following will be received by the daughter cells after the cell division to ensure proper working of cells? Okay, cytoplasm, Golgi apparatus, endoplasmic reticulum, DNA, exactly. Okay, for the proper working of daughter cells, ultimately what should be transferred to the daughter cells? DNA, because DNA is the genetic material. Okay, yes. Now, which of the following is under the genetic control? DNA synthesis, cell cycle, distribution of genetic content, all of the above, and interestingly, all of the above is the answer. Okay, so now, till now, whatever we have discussed about the interphase and also the M phase, or we can say complete cell cycle, the whole cell cycle process itself is under the genetic control. Remember, okay, the genetic control, it is controlling every activity of the cell, including the cell cycle. Remember, okay, even the cell cycle is under the genetic control. Yes. The time required for the mammalian cells in the cell cycle, I mentioned, I forgot the point. The time required for mammalian cells to divide, let me write this. divide is 38 hours, 24 hours, 48 hours, 15 hours. We know it is what? The 24 hours. The phase of the cell cycle which actually cell divides. Okay, so let's look into the question. The phase of the cell cycle in which a cell actually divides is the M phase, okay, interphase, S phase and G phase. The G1 phase and S phase are actually part of the interphase, and we know interphase is a phase where cell does not divide. It is actually the M phase where the cell actually divides. The next one. During the cell cycle, the mammalian cell consume a major of its time in a G1 phase, S phase, G2 phase, all of the above. The answer is again all of the above, because ultimately all of these three stages make up the main phase called as the interphase. Okay, so don't expect only interphase the option to be present. Okay, they may even give you all the three options and they can ask you all of the above. Okay, that is also possible. So we know G1 phase, S phase, G2 phase, they're actually part of the interphase. Hence, all of the three make up the interphase, and indirectly we can say that the cell is spending all of its time in these three phases for a majority movement only. Right? And we know that it spends mainly around 95%. Yes, around 95%. Just a moment to recall. So now, at any moment during the cell cycle, if the materials for the next phase of cell cycle is not produced, then the cell cycle continues, cell division is terminated, only nuclear division occurs, none of the above. The answer is the whole cell division process is terminated. Okay, so what happens is that if the nutrients that are required for the S phase, if they are not produced, then how can the DNA replication occur in the S phase? It is not going to occur. So we can say that it is terminated, right? Yes. So similarly, in the case of G2 phase, whatever nutrients is required for the, or molecules required for the M phase, if it is not produced, then what happened? The cell will not divide only. Yes. So we can say that the whole process of cell division itself is terminated. Hence we can say B is our right answer. The next question. In embryo cells, in embryo, the cells constantly divide through a series of mitotic division without the increase in the size of cell. This is due to fast cell division time, no S phase, no gap phase, and no interphase. Okay. Now, this is an important point that we can discuss over here. See, remember, in the case of embryo, we are having a special kind of cell division, mitotic cell division, series of cell division called as the cleavage. Okay, it is called as the cleavage. In the case of cleavage, as we said, it is also a kind of mitotic division only. But however, a single cell divides into 2 cell and 2 into what? 4 cell and 4 to and so on, right? Yes. But interestingly, during the cleavage, there is no increase in the cell size. Okay, no increase in the cell volume. But in the normal cell division mitosis, you see that there is increase in the cell volume. Now this is mainly due to that in the case of cleavage mitotic division, there is no gap phase. We can say that from S phase to M phase, and from M phase to directly S phase. There is no gap phase. Hence there is no increase in the cell size. So within the small tiny amount of tiny cell that is present, the number of cell gets added on. So we can say the right answer for here is there is no gap phases, no G1 phase, no G2 phase, only from S to M and M phase to S phase directly. Yes. Which of the following is not a characteristic of the M phase? Okay, that is cytokinesis, DNA replication, and karyokinesis, spindle fiber formation. So spindle fiber actually is mainly required for splitting of chromosomes, and that is actually formed in the M phase. We can see that cytokinesis is actually a part of the M phase, and DNA replication is actually part of the S phase, and in the case of S phase you can see that it is actually part of the interphase. Okay, it's part of the interphase. Yes, exactly. So you can guess that the right answer is going to be the B, that is DNA replication is not a characteristic of M phase. Next, the inhibition of mitotic spindle in the M phase will result in inhibition of chromosome formation, inhibition of interphase, inhibition of chromosome arrangement in the equatorial plane of the cell, inhibition of sister chromatid separation. Okay, so you can see that the mitotic spindle is mainly required for the separation of sister chromatids from the chromosome. So if I am not allowing the mitotic spindle to form, you can say that there is no separation, or we can say inhibition of the what we can say sister chromatid separation. Okay, rather than the term inhibition, we can say that no sister chromatid separation is observed. So the more appropriate answer for this question we are having is the D, that is inhibition in the separation of sister chromatids. Okay, that is the event which is more directly dependent on the spindle cells itself. The next question: during the S phase, the chromosome number increases, DNA duplication occurs, mitotic spindle forms, centriole duplicates. So it's a very easy question. S phase is mainly involved in the DNA replication. Yes, B is going to be your right answer. The next one: the cell cycle events are under the control of DNA, RNA, proteins, ribosomes. It's an easy question. It is mainly your DNA. It's a genetically controlled process. The next one: the time the yeast cells require to complete the cell cycle is 1 hour, 30 minutes, 1.5 hours, 20 minutes. We know it is around 1.5 hours or around 90 minutes exactly, right? Yes. So 20 minutes is mainly in the case of bacteria, remember. Okay, in the case of bacteria it is around 20 minutes, the cell division or cell cycle, the whole process of cell division we can say. Now the 16th question: during which phase the cell grows and a nutrient accumulation is observed? Okay, the gap phase, the synthesis phase, mitotic phase, quiescent phase. We all know it is mainly the gap phase where the nutrient accumulation and cell actually grows. The next question: the number of chromosomes present in each cell of onion is exactly. Now you people will be doing this as a practical in your second year examination, that is a squash mitotic, that root squash preparation, right? So the number of cells that are found in the case of a diploid onion cell is actually around 16. Remember this, the diploid number is 16, but however haploid is 8. This is actually the haploid you can say. Yes. The next one: any cells that are metabolically active and quit the cell division and can enter back to cell division when required are present in which phase? That is a G1 phase, S phase, G2 phase, G0 phase. We know it is actually the G0 phase, as we have discussed about the G0 phase earlier. The next one: which of the following cell have the ability to return from a G0 phase back to actively dividing phase of the cell cycle? Stem cells, kidney cells, nerve cells, and heart cells. So I have given an example. It is mainly the stem cells that have the ability to come back from G0 phase to active dividing. Okay, but however kidney cells, they are completely differentiated. They don't go back from G0 to cell division stage. Okay, yes, as of now what I know, kidney cells don't go, but nerve cells and heart cells, they never go itself. We know this. The next one: haploid cells of dash show the mitosis. So humans, bats, honeybees, and amphibians, it's an easy question. It is mainly the honeybees that show, we can say, the mitosis. Haploid cells of the honeybees show the mitosis. So C is our right answer. So students, we have discussed around 20 questions in the first class. And as I said, it is going to be a very short class. You don't have to, we can cover this chapter in a very short period of time. But however, this chapter is quite important because some of the questions, especially on the S phase, about mainly the genome content and all, you can expect. Okay, yes. So we will meet in the next class of the cell division and cell cycle chapter, and you people can start solving the questions on this particular part through the material that you people are having. Okay, refer any material, don't worry. Okay, and you can try to explore high level concepts if you want to, so right. So we will meet in the next class. Till then, goodbye.